Now, while many disinfectants are not used to kill microbes in or on the human body, there is a surefire way, or at least it used to be, to kill bacteria that are causing an infection in human tissues. And this is antibiotics. Antibiotics are chemicals that are produced by one microorganism to kill other microorganisms. They have been around for arguably billions of years as a result of microbes competing with each other in the soil. This soil competition caused one microbe to generate a chemical compound that would kill other microbes, their competitors, and allow the first microbe to take control of the area and all of the food and water in that space. In turn, the other microbes in the soil evolved resistance, or they died. But the ones that were able to stay alive evolved means to resist the antibiotics. So antibiotic resistance has also been around for billions of years. The difference is that the antibiotics are now used to kill human pathogens, not bacteria in the soil. And as a result, human pathogens are evolving the ability to resist these antibiotics. Bacteria vary in their ability to resist or be susceptible to antibiotics, and each antibiotic has a spectrum of activity against certain bacteria. A broad-spectrum antibiotic is one in which it kills a large number of gram-positive and gram-negative bacteria. Narrow-spectrum antibiotics can only kill a limited range of bacteria. In addition, another component to always consider is that some antibiotics can be toxic to some human tissues or cells. While some antibiotics are toxic to some tissues, other antibiotics can generate allergies. This is particularly true of penicillin and sulfonamides, or sulfa drugs, though all antibiotics can exhibit some form of allergy in someone. Some antibiotics are better getting to some parts of the body than others, like kidneys or brain tissue. And finally, some antibiotics stay in the body longer than others do, allowing them to be more effective over a longer period of time. Antibiotics also differ in how expensive they are. And so some antibiotics may be passed over just because they're more expensive to be produced and purchased. Broad-spectrum antibiotics are often used more regularly than narrow-spectrum, because in order to use a narrow-spectrum antibiotic, you must definitively diagnose what is causing the infection. However, broad-spectrum antibiotics are used when the cause of the infection is not fully known. The downside, of course, of broad-spectrum antibiotics is that a large number of bacterial species of your normal microflora or microbiota are killed. This causes an ecological disruption of your normal microbiota on the skin, but primarily this is noticeable in the large intestine. When this happens, people can end up getting diarrhea or not feeling well, which is why we don't want to prescribe antibiotics when they're not necessary. Normal microflora of the vagina and of the oral cavity or mouth can also be disrupted and result in yeast infections. Yeast are not killed by antibiotics because they are fungi and not bacteria. So when your normal bacterial communities are destroyed in your mouth or vagina, that can allow the yeast to grow out of control and generate disease, such as in the case of vaginal yeast infections or a similar yeast infection in the mouth called oral thrush. Next, we'll take a look at the mechanisms of action for these antibiotics. Antibiotic drug targets are going to have an effect on different components of microbes that human cells do not have. This is what's known as selection or selective toxicity. That essentially means that the drug is going to kill the microbe or be toxic to the microbe, but not to human cells. One of these really good targets is the cell wall of bacteria. Human cells don't have a cell wall at all, whereas nearly every bacterial cell has a cell wall made of peptidoglycan. Many of our best antibiotics target the peptidoglycan cell wall of bacteria. This includes penicillin and cephalosporins. Cephalosporins actually account for about 1/3 of all antibiotics used today. Vancomycin also targets the cell wall of bacteria, but because it is highly toxic to the human cells, it is not regularly used. Other antibiotics interfere with protein synthesis. This includes aminoglycosides like streptomycin and gentamicin, chloramphenicol, which is not regularly used in hospitals but is frequently used in research labs, tetracycline, and erythromycin. All of these drugs target the function of ribosomes. Ribosomes are the cellular enzyme complex that's responsible for taking the message found in RNA and turning that information into a protein. By targeting the ribosome, these antibiotics prevent protein synthesis. Remember, proteins are the function molecule of the cell. They are required for the cell to do anything, including grow, take in food, remove waste, swim, or invade host tissues. Without protein synthesis, the cell cannot do any of these things, and thus the cell would die. Interestingly, human cells have a different type of ribosome. All cells require ribosomes, but bacteria have different ribosomes than eukaryotic cells do, and thus selective toxicity is also held with these antibiotics. However, our mitochondria, or the organelle inside our cells that's responsible for making ATP, do have the same type of ribosome because they are essentially little bacteria living inside of our cells. Because they have the same ribosome, some of these antibiotics mentioned here can be effective against our mitochondria and cause diseases such as anemia, fatigue, and other issues with our own cells. Less common are antibiotics that are used to interfere with cell membranes. The reason for this is, of course, that we have cell membranes as well. In fact, our cell membranes are very similar to bacterial cell membranes. Cell membranes are essential for all cells because they act as gatekeepers, keeping vital and important molecules inside the cell and preventing them from leaving, and keeping toxins outside the cell. Without the cell membrane, toxins can come in and important molecules like DNA can leave. This can end up killing the cell. Next, antibiotics can also interfere with nucleic acid synthesis. This is the formation of RNA and DNA. Quinolones are one example of this. They're involved in targeting DNA replication. There are other antibiotics that can target RNA synthesis, especially by the enzyme RNA polymerase. Our last category are antibiotics that affect metabolic reactions. These include those sulfonamides that I mentioned earlier, otherwise known as sulfa drugs. Remember that these are ones that are associated with high levels of allergies, including penicillins. Sulfon drugs, or sulfonamides, affect and target the formation of folic acid. Bacteria make their own folic acid from food that they eat, and they make it using enzymes that produce the folic acid. The sulfa drugs target these enzymes and keep them from functioning. Folic acid is important because it's used to generate nucleotides, such as found in DNA and RNA. Again, this works by selection, or selective toxicity, because human cells obtain folic acid or folate from our food. If you've known a pregnant woman, she is likely needed to take prenatal vitamins that contain additional sources of folate because our food does not always supply pregnant women with as much folate as they need. So we do not synthesize folic acid, and thereby making the sulfa drugs, or sulfonamides, completely ineffective in human cells. There are some challenges to using antibiotics. Nearly all of the antibiotics that we use in clinical medicine today target a microbial process. This means that when the bacteria are growing more slowly, the antibiotic does not have as many opportunities to act. For example, if an antibiotic acts on a cell wall when the organism is growing slowly, it will take longer for the antibiotic to result in cell wall weak enough to lead to bacterial cell lysis. Some bacteria naturally grow slowly, which means the antibiotic will need to be consistently taken for a long time to treat that infection. This is what happens with Mycobacterium tuberculosis, the bacterium responsible for causing tuberculosis. It usually takes about 6 to 9 months of antibiotics in order to treat this disease. Antibiotic susceptibility testing in the lab also takes a long time because it takes so long for the bacteria to grow on Petri plates. By the time the results show what antibiotics the bacterium can resist, the patient may have been on the wrong antibiotic for weeks. Another challenge in the treatment of an infection is that the antibiotic may not have easy access to the bacteria in the patient. This is especially true for infections that have formed a biofilm. Biofilms are microbial communities that stick together on surfaces of foreign objects or unhealthy body tissues. This could be catheters, pacemakers, or in the case of a patient with cystic fibrosis, their own lung tissue. As the biofilm persists, clumps of the biofilm which contain bacteria can break off and allow an infection to persist and spread to other sites of the body. Bacteria that are deep within the biofilm may be metabolically inactive or dormant, and the antibiotics that target growth are ineffective on these microbes. Additionally, the antibiotic may not even be able to penetrate the biofilm through the surface layers in order to gain access to the bacteria within. As a result, if the biofilm forms on a medical device, such as a catheter or a pacemaker, the device is often physically removed and replaced with a new one. In the case of cystic fibrosis patients, there isn't much that can be done, except for possibly lung transplants. Another challenge in the clinical setting is the access of the antibiotic to growing bacteria. This can happen if there's an infection in bone marrow, where very little blood is transmitted to the bone marrow itself, keeping antibiotics from reaching the bone infections. Infections of the central nervous system also need antibiotic drugs that can cross the blood-brain barrier, which is an effective barrier keeping many molecules out. In the next section of this chapter, we'll talk about the acquisition of antibiotic resistance.